Now in this module, we shall learn the different methods for measuring the refractive index of the glass which is under crime investigation in details. In our previous module, we had studied these methods only in brief, and now we will study them in details. But in the previous modules, till now what we've understood is that one of the most important problems in forensic glass analysis is the comparison between the known and the unknown glass samples in order to establish their origin. And for that purpose, various analyses have been done. Now, till now, we have studied only the density measurements in details, and now we are going to study the refractive index measurements. This module is actually in continuation with the first part of the refractive index module, in which we have already studied the apparatus, or in other words the instruments that we are going to use in the analysis of the glass fragments and the methods we are going to adopt only in brief. Now we are going to do them in details. Now this refractive index measurement holds importance in forensics because it actually gives us a hint about the make and the history of the glass, and thus it makes us possible to do the analysis properly in details. But it is not actually possible to measure the refractive index directly. The reason being that it is not possible to practically measure the speed of light when it travels through the glass. As a result, what we do is we adopt some indirect methods. Some of the methods which we are going to discuss in this module include Emmons double variation method, which is a very famous method, Backline method, automated glass refractive index measurement, which is nothing but the automatic measurement of the refractive index. The last one is going to be the dispersion staining. Now, whichever method we adopt, the most important feature of these methods is that we make use of the principle of refraction and dispersion. So basically, these methods are based on the fact that if these glass fragments under investigation have got same refractive index as the liquid in which it is immersed, then the glass fragments become invisible, and thus we can actually note down the refractive index of the glass. Let us now start with Emmons double variation method in slight details. Now, the most common and the oldest method to measure the refractive index of the glass till date had been Emmons double variation method. This method was actually developed by Richard Conrad Emmons at the University of Wisconsin circa in 1928. He actually found out that by manipulation of both the temperature and the wavelength of light, it was actually possible to obtain not only the refractive index of the glass, but also how the refractive index varies with the wavelength of the light that is being used in this method. So basically in this method, the slide containing the glass fragments was immersed in silicone oil. Here, the oil or the liquid was chosen in such a manner that the temperature coefficient of the refractive index, that is denoted by dN by dt, must be of the order of 10 raised to power minus 4. Also, it must be stable within the temperature range that they were going to consider for this method. This slide with the liquid is then inserted into the so-called hot stage and is then illuminated using a monochromatic sodium light with wavelength 5890 angstroms. The temperature of the hot stage could actually be controlled for heating as well as for cooling within a precision of almost 0.2 degrees Celsius or even better. For taking the observations, a phase contrast microscope was then used. A phase contrast microscope, basically, as we have already studied in the last module, converts actually the phase shift in light passing through the transparent specimen to the brightness changes in the image. For noting down the observations, the refractive index of the oil was kept on changing by varying the temperature of the oil mount and also the wavelength of the light used for this method. Until the backline disappeared and there was a minimum contrast between the glass and the liquid medium. Now, the temperature at which the backline disappears is considered to be the match point at which the refractive index of the glass actually equals the refractive index of the silicon oil. Now, there are some advantages along with some limitations of this method, which are as follows. Among the advantages, now the wide variations in the refractive index of the oil, it actually reduces the number of oil mounts needed. Now, this is advantageous as a change in the oil involved can damage the small grains itself after the match point is obtained. Now, this method provides a very rapid means of measuring the refractive index of glass at multiple wavelengths also. Now, the precision of this experiment is almost from 0.0004 to 0.00006, which is very good. But apart from these advantages, there is also one limitation. That is, this method will actually not be able to differentiate between glass samples from different sources with refractive index less than 0.0001, which is again a disadvantage. Now then, for that purpose, automated refractive index measurements were done. Now, Emmons double variation method, it's still in use in some of the laboratories, but it has actually been completely replaced since the 1980s by an automated method which is referred to as GRIM. It is known as glass refractive index machine due to the model name which is given by the company which produces the instrument. Now, the automated determination of refractive index using oil immersion method along with the phase contrast microscope and a video camera can be done for glass samples as small as 300 micrograms and is called ASTM. Now, in this method, the glass fragments should be cleaned carefully with deionized water and ethanol, then dried and then crushed in very small pieces of glass of approximately 1.5 micrometer in size. Now a sample is then deposited on a slide and immersed in a micro drop of oil and covered with a cover slip. In this method also, the selected immersion oil or liquid should have temperature coefficient of the order of 10 raised to the power minus 4. Also, the oil must be stable across the temperature range under investigation or under consideration. The slide is put on the hot stage then. Here, the temperature of the hot stage is controlled by GRIM fitted with a contrast microscope and a video camera. The temperature of the glass is changed in a very controlled manner by using the hot stage. Now, the temperature at which the glass disappears, that is there, is a minimum contrast between the oil and the glass. It is then recorded electronically, and an average of the match from both the heating and the cooling cycle is converted to refractive index value according to the calibration curve of the oil, which is known to us. In detail, this means the temperature of the hot stage is adjusted in such a manner that the refractive index of the liquid is initially higher than the sample, but during the analysis, the instrument lowers down the temperature of the preparation or the hot stage through the match point for the glass. The contrast between the glass fragments and the liquid is then observed and monitored, and then the matching point is noted down. This procedure is then repeated as the temperature is now raised through the match point. These values are then recorded as the match temperature on cooling and the match temperature on heating, which are then averaged over to give the match point temperature of the sample. This is, as discussed earlier, is converted to the refractive index value according to the calibration curve. The calibration curve has been obtained by standardization of oils using reference glass with certified values of the refractive index. ASTM recommends a minimum of 3 standard glasses for the calibration of each oil. Now, in this method also, there are some advantages as well as some limitations. Advantages are as follows. Now, the automated method for measuring of refractive index has the basic advantage of improved precision as well as improved accuracy. Now, the refractive index is automatically generated here in this method and electronically recorded. Over the years, GRIM1 has now been replaced by GRIM2 and then by GRIM3, which is the latest available. But apart from this, there are 2 limitations also as follows. The hot stage, the glass slides and the microscope must be very clean. Traces of liquid transferred from glass slides to the inner surface of the hot stage can give anomalous results. Now, this method will again not differentiate between glasses with a refractive index difference of less than 0.0003. Then comes the Backline method. Now, Backline method is also one of the most important methods for determining the refractive index of a transparent particle relative to its surrounding medium. Now, the advantage of this method is that it not only indicates the difference between the refractive index of the glass and the liquid, but also indicates which one possesses the higher value. We know that when an object is immersed in a liquid, a bright fringe called backline is observed at the object interface. Now the glass fragment under investigation is immersed in a liquid which has got known refractive index. Now the difference between the refractive index of the glass fragment relative to the liquid is then observed. Now in this method, a bright halo or a backline is observed. The movement of this backline with respect to the object or the fragment on changing the focus of the microscope then indicates the refractive index of the object relative to the immersion oil, that is, whether the refractive index of the glass is greater than or less than that of the liquid. Now the difference in the refractive index is indicated by the amount of contrast between the glass and the immersion liquid. The fragment is then removed, washed and placed in another liquid, again with the refractive index which is closer to the match point. This is repeated until the refractive index of the match point has been deduced, that is, by using a succession of liquids with different refractive indices, the refractive index of the sample can be obtained. Now again, this method will also have some advantages and disadvantages. Now some of its advantages are that it requires only one microscope, calibrated liquids, the means to calibrate them, that is a refractometer. It helps in rapid sorting out of very different glass sources. But it also has got some limitations which are as follows. Now the accuracy of the backline method is actually limited by the visibility of backline. Factors such as object roughness, object inhomogeneity and small object size diminish the visibility of the backline and the accuracy of this technique. It actually becomes difficult to recover the glass fragment under investigation and then to clean it properly between liquids. As a result, evidence can be easily lost or liquids may get mixed up, resulting in errors in the measured indices. Now, also the results can be reported only within the accuracy of 0.001. Thus, it does not form a good discrimination between the different sources. Also, the refractive index of the liquids vary with the temperature of the liquid. So, the laboratory temperature has to be maintained at a fixed value, else some error can be introduced in the system. Now coming to the last method of dispersion staining. By dispersion we actually means the splitting up of a visible light into different colors. This is because of the fact that different colors bend with different diffracting angles as they have got different wavelengths. Dispersion curve is nothing but a graph between the changes in the refractive index to the wavelength. Now the dispersion staining method is based on the principle of dispersion. It is an analytical technique that takes advantage of the difference in the dispersion curve of the refractive index of an unknown material relative to a standard material with a known dispersion curve to identify the unknown sample. Now these differences become manifest as a color when two dispersion curves will intersect for some visible wavelength. Now it is an optical staining technique and does not require any kind of dyes or stains to produce the color. Now, when the conditions for dispersion staining are met, that is, once the glass is mounted in a liquid with a matching refractive index in the visible range, but with a very different refractive index, then the glass has a high refractive index in the red part of the spectrum and a lower refractive index in the blue part of the spectrum. Now, liquids, they tend to have a steeper dispersion curve. The dispersion staining color is converted into refractive index value, which is then added or subtracted to the refractive index value of the glass, standardized to obtain the refractive index of the liquid. Now, dispersion technique being an optical effect and not involving a chemical reaction, it can thus be widely used in the study of everything from dental cares to hazardous material identification. The most common application is the analysis of the asbestos fibers, but it has many other useful applications. Now, there are 5 types of dispersion staining techniques, or in other words, there are 5 different optical configurations for the microscope which is used for dispersion staining method. These configurations are as follows: backline dispersion staining, oblique illumination dispersion staining, dark field dispersion staining, objective stop dispersion staining, phase contrast dispersion staining. Now, each configuration has got its own advantage and disadvantage. Out of these techniques, backline dispersion staining and oblique illumination dispersion techniques were first reported in the United States, first by F.E. Wright in 1911. All these have the same requirements for the preparation of the sample to be examined. That is, firstly, the glass sample under question should be cleaned and should be in proper contact with the known reference material. In other words, this clean glass sample should be mounted in a reference liquid. Now, dispersion colors will present only if the two materials have the same refractive index for at least some wavelengths in the visible spectrum, but have different dispersion curves for the refractive index, as we have already discussed. Now, a brief sketch of all these techniques is as follows. Now, backline dispersion staining is based on the fact that the particles, they behave like lens when immersed in a liquid, as they tend to be thinner at the edges than the center. Now, if the refractive index of the particle is greater than the surrounding liquid, then it behaves like a convex lens and will focus a parallel beam of light on the side opposite to the source of the light. This, under microscope, is seen as a bright ring of light called backline and moves in from the edges as the particle is dropped out of focus by increasing the distance between the stage of the microscope and the objective. If the stage is further moved closer to the object, then the particle will behave as a magnifying glass and the magnified image of the backline now appears outside the particle. So, the most important requirement for the method is that the incoming beam of the light should be made parallel. As we have already discussed, once the conditions are met, then the particle has a refractive index higher in the red part of the spectrum while lower in the blue part. So, for the blue wavelength, now the particles behave like a concave lens and the blue backline moves out of the liquid. The color of these two bands will vary depending on the particle and the liquid match in a refractive index. For example, if the match is near the blue end of the spectrum, then the backline moving into the particle contains the entire visible wavelength except blue and will then appear pale yellow, whereas backline moving out will now appear dark blue. But if the match is near the red end of the spectrum, then the backline moving into will appear dark, while moving out will be pale blue. For larger particle, this is actually one of the best methods. Now coming to the oblique illumination dispersion staining. In this technique, the beam of the light illuminating the sample is made to fall at an oblique angle through the sample. Although this will enhance the resolution of the structural details in the particle oriented at right angles to the incident beam, while some of the resolution of the features parallel to the direction of beam are then lost. As a result of this orientation, the relative refractive index of the particle and the surrounding liquid becomes apparent. So, the wavelength for which the refractive index takes a higher value, they are refracted into the front lens of the objective from the side of the particle nearest to the side from which the light is incident. Thus, if the particle has a higher refractive index for all the visible wavelengths, then this side will appear dark, while the side farthest from the source of the light will show all the wavelengths. Coming to the dark field illumination dispersion staining. Now, this type of dispersion staining is actually due to the image of the particle which is formed by the refractive in light, while the direct light incident on the sample is oriented at such an angle that it misses the front lens of the objective. The result is that the background is now black, while all the features of the objects in the field of view that don't match the refractive index of the medium will now appear white. Now, when a particle is mounted in a liquid that matches the refractive index within the visible range of wavelength, then those which are refracted by the particle and are thus not collected by the objective. So, the image of the object is formed by the remaining wavelengths then. These wavelengths will combine to form a single color which is used to indicate which band of wavelength is missing. Now, coming to the phase contrast dispersion staining. Now, this technique will require a phase contrast objective. That is, it takes into consideration the fact that the rays of light that are not shifted in phase by the presence of the object are separated from the light rays which are shifted in phase at the back focal plane of the objective. Now, these unaffected rays are actually significantly decreased in intensity. The particle will now appear colored from the contributing wavelengths for which the mounting medium has the higher refractive index. Coming to the last one, which is objective stop dispersion staining. This type is based on the fact that all the light unaltered by the presence of particles in the field of view is then focused at the back focal plane of the objective. This method enhances the color effect but loses the resolution of the particles. Now, it has got also some advantages, that is, this method has the advantage as the Backline method, that is, it requires only one microscope, calibrated liquid, refractometer and a dispersion staining objective, and provides rapid sorting of different classes. The limitations of this method is that very extensive training in the use of dispersion staining method is desirable for this technique, since the reliability of the results is quite dependent on the skills of the analyst. In this technique also, the results can be reported only within the certainty of 0.001 even under the best conditions. Now, coming to the summary of this module. In this module, what we have studied is the various methods of determining the refractive index of the glass fragments under investigation. This measurement actually makes it very easy for the forensic investigators to know the history and the make of the glass, which can actually serve as an important evidence of this crime which is under investigation. Now, since the refractive index cannot be measured directly, it has to be measured using some indirect methods, which include Emmons double variation method, automated refractive index measurements, Backline method and dispersion staining techniques. All the above methods are based on the principle of refraction and dispersion, that is how light bends when it passes from one medium to another, and the splitting up of light into its 7 colors. In fact, there are further 5 types of dispersion techniques.